Before samples can be sent out for sequencing, they have to be first normalized. In order to perform the normalization step, we have to transfer the second step PCR product to a 96 well plate, which then will be processed on our pipetting robot. The normalization procedure has to be set up in the robot software beforehand. And here it is, the pipetting robot, a great lab facilitator. Thanks to this device, one can greatly enhance the reliability of the pipetting steps, as well as save time and perform or prepare other steps of the analysis while the robot is running. The normalization procedure is an important step when preparing a metabarcoding library. By normalizing the samples, we're making sure that all our samples have similar DNA concentration and are equally represented in the pool. Thanks to that, we are avoiding an overrepresentation of single samples. In our case, the normalization is performed using a dedicated normalization plate, where each well has a special coating allowing to bind DNA at the desired concentration and length. With that, we are making sure that we are excluding the primer sequences from the library. Afterwards, since all the samples are now normalized, we are ready to pull them together in a single library. Don't forget about the proper labeling. Before removing the sealing foil from the PCR plate, always check if there are no droplets of liquid on the foil. You're not supposed to touch the inside of the foil nor the well's openings. It is good practice to fold the foil before discarding. To create a final sequencing library, we are now pooling every single sample together. Since they are all tagged differently, we will be able to retrieve the DNA signal from each of the samples separately, even though they are now pooled together. This is the final product of our entire lab processing. Even though it does not look like much, it is containing all the relevant DNA information from 2 sampled sites, including both sorted and unsorted bulk samples, as well as the eDNA information. Here we also have performed a column cleanup of our library to enhance the DNA concentration. To ensure that the normalization worked properly and we have primarily our targeted fragment of DNA, we are checking the library using the so-called fragment analyzer. In fact, one could say that we are now running a really fancy gel electrophoresis. But comparably, it will deliver much more detailed quantification of our library than an agarose gel. When the results obtained through the fragment analyzer indicate high amount of targeted DNA fragments, which in our case is 611 base per fragment, as well as sufficient DNA concentrations, we can then prepare our library for sending. When packing a library, it is best to seal the tube containing our library with arguably the best way possible, with parafilm, the most satisfactory lab fabric ever. It is also good to remember about adding a frozen cooling pad, providing the right temperature for our sample during transport. Now we are finally ready to send our samples for sequencing. Fingers crossed.